How do you say your name? Frietze? Frietze?
Fritz, yep.
Fritz. Seth Frietze, University of Vermont. Uh, Seth is a assistant professor in the Department of Biomedical and Health Sciences at UVM. He recognizes that whole genome sequencing applications have created an incredible revolution in the way we study disease and biology. And the research in his lab applies these approaches to study the molecular mechanisms of gene regulation with long-term goal to discovering new therapeutic strategies to target cancer cells. So
And
today,
is
he's going to
an
talk about.
associate professor in the Department of Biomedical and Health Sciences.
Oh, is the associate? Okay,
Yes.
gotcha. Yeah, yeah, yeah. All right. Yeah, associate professor. We'll give you a boost here. Uh, his pilot project today is entitled Characterization of CDK12 as a Breast Cancer Therapeutic Target. So, Seth, take it away. It's all
Great.
on you. Yeah.
Thanks for the introduction, Rob, and thanks for everybody to be here today. I'm really happy to have this opportunity, and I'll dive right into it. Yes, I'm an associate professor here at UVM, and I'm also a program co-leader of the UVM Cancer Center Cancer Cell Program at the UVM Cancer Center. Um, and I just want to acknowledge the NNE-CTR, and I'm really grateful for these working with this group. It's really important to provide these opportunities as us basic scientists, and to have these resources to help support some of these clinical and translational projects that we have. And the project I'm going to tell you today is really a very basic project, but I hope that if we can continue to develop it, it might help lead into something more valuable than our understanding of basic cell mechanisms. And here at UVM, I'm really grateful to have a strong team. And so this project is really driven largely through work together with the Center for Biomedical Shared Resources at UVM, and a collaboration with Peter Kaufman, a breast cancer oncologist here at UVM, and a very talented postdoc in my laboratory who's has an expertise in cyclin-dependent kinases, which is what I'm going to be talking about. And of course, these pilot projects have really helped so much. So I'm going to just jump right into the pilot project. This proposal sought to investigate this one protein called CDK12 as a therapeutic target, and specifically in HER2 positive breast cancer. And we had some pretty lofty goals with this project. We proposed two aims. One, let's use some biochemistry and some mass spectrometry to identify functional regulators of CDK12, with the overall hypothesis that if we could understand more about this protein, we might find ways to intervene with it in cancer. And then aim two was to establish a cell model where we can study the CDK12 dependent mechanisms and learn more about what it does in cancer and cancer cells, to have some mechanism with this possibility. So by way of background, breast cancer is a heterogeneous disease. It's not just one disease, it's many different types of diseases. And classically, this can be determined, this disease heterogeneity can be classified by immunohistochemistry and these pathological markers such as the estrogen receptor or progesterone receptor, these hormone receptors. And indeed, the majority of breast cancer patients express these proteins and are classified in this group. And then a sizable number of these patients don't express either of these proteins. These are so-called hormone negative tumors. And among this group are the HER2 positive tumors, a biomarker for epidermal growth factor protein on the cell surface, and then the triple negative breast cancer class that do not express either of these receptors. And so, in the advances in the turn of the around early 2000s, in gene expression analysis, classified these tumors into different groups, and associated with these molecular subtypes of cancer were clinically relevant measures of outpatient outcome. And from these, we can see that the groups, the ERBB2 or so-called HER2 positive group, or these triple negative basal-like groups, represent quite aggressive tumors that are associated with poor outcome. Targeted therapies exist for the HER2 positive cancers, but we don't have any targeted therapies for triple negative breast cancer. So HER2 positive breast cancers are classified by expression of this HER2 protein on the cell surface shown here in this staining of a specimen, and then due to the biology and the transformation, you have this proto-oncogene becoming an oncogene through amplification typically. And so these cancers tend to have many copies of this region of chromosome 17, the so-called ERBB2 amplicon. It's a large section of DNA that gets highly amplified in many of these tumors, resulting in overexpression of this HER2 protein. It's a plasma membrane protein that serves to dimerize with other receptors and transmit growth signaling into the cell that promote tumor proliferation. And this classification of this tumor is directly related to the targeted treatment, in that antibodies such as I always have a hard time, Herceptin, target this cell surface receptor and block the cell growth. And other advances has been development of tyrosine kinase inhibitors that are able to block downstream signaling. And so together, these treatments have really revolutionized the care of HER2 positive breast cancer. But with these targeted therapies, resistance still remains a major problem, and we need new targets. So one of the benefits of being at UVM and participating at UVM Cancer Center and participating with some of what we call them as transdisciplinary teams, one of them is genomic medicines, is you get to hear about all these cool clinical trials and reading about getting connection with the clinical aspect of this science. And it's clear that cyclin and cyclin-dependent kinases are major targets in cancer treatments, and a lot of efforts are underway to develop specific inhibitors that block cyclin-dependent kinase processes, because they're so important for cell proliferation and cell division. If we took all the cyclin-dependent kinases and looked at them like this, we'd see that there's many of them, and a lot of them share sort of similarities in that they have not a lot of action going on the primary protein level, largely a kinase domain, with the exception of these two transcription-related cyclin-dependent kinases, CDK12 and CDK13, which are considerably bigger than most cyclin-dependent kinases, and they also have additional protein domains such as arginine, serine-rich domains commonly associated with splicing factors, and these proline-rich motifs. And like all cyclins, these proteins form obligatory partners with the cyclins that control their activity and cell cycles expression. Although these transcriptional kinases seem to be less directly cell cycle regulated, more just involved with fundamental mechanisms of cells. In HER2 positive breast cancers, I mentioned the ERBB2 is the gene that encodes the HER2 oncogene. And in this amplicon are many other genes. And so CDK12 is one such cyclin that's commonly, it's neighboring ERBB2. So when ERBB2 is amplified, CDK12 expression is high. And this study did a nice job analyzing Metabric and TCGA data to show that many of these genes associated with this HER2 amplification on this amplicon were strongly associated with disease-free survival and overall survival. And that CDK12 in particular is a cyclin-dependent kinase in HER2 positive breast cancer, where the copy number is highly correlated with gene expression. It seems to be much higher expressed in HER2 positive versus the hormone or triple negative breast cancers, and patients that have high expression of CDK12 in these cohorts have a overall poor survival, similar to the ERBB2 expression. So what does CDK12 do? I mentioned it's a transcriptional kinase. It's got known functions in central biological processes. Most notably, it's a RNA polymerase II kinase. It phosphorylates this protein that's important for gene transcription, and it regulates its activity. And it's not very well understood how it works or what its roles are in this process, except that it's important. And if you target it, you can really mess with cancer cell biology. And so we sought out, our first aim was to identify CDK12 interactomes, to study its protein-protein interactions, to learn more about its function, and its function in particularly in HER2 breast cancer cell lines. So to do this, we took a protocol called proximity biotinylation. What this means is we generate a cDNA vector that expresses the CDK12 gene inside of cell lines of interest. We generated this system using both an N-terminal and a C-terminal fusion to this protein called mini-turbo. Mini-turbo is an enzyme. The way it works inside of cells, when it makes CDK12 fused to this enzyme, shown here as BRA, turbo is a version of this BRA enzyme that works faster to label. And the way it works is that cells make this protein, you can feed the cells biotin, and then proteins within any proximity of this enzyme, aka CDK12 interactions, get biotinylated. And then we can break open the cells. We can use streptavidin beads to purify biotinylated proteins, and then mass spectrometry to identify CDK12 interactions for both the N and C terminus. We did this in HER2 positive cell lines, number of cell lines, and excuse me? Oh, sorry. Including SKBR3 cell lines that contain both the CDK12 ERBB2 amplicon. And so here's some of the results from this study, from this pilot project that's worked quite well, and I'll walk you through some of these results briefly. What this is showing is cell lines, breast cancer cell lines, that express a control, untagged turbo-only enzyme, or of different variants of CDK12. We have this N-terminal short isoform. We have a C-terminal short isoform and a C-terminal long isoform. And we also tagged its obligate partner, CCNK, the cyclin K gene, both N and C terminus. And when we express these in cells, we can see an increase in CDK12 with this CDK12 Western blot. The cells, this is a pilot, right? We got to learn optimized conditions, and it turns out that cells like this N-terminal cyclin K more than the C-terminal cyclin K, and we can see this results in more robust expression. This turbo ID protein also has an HA epitope tag that we can use to detect it. And so we see good expression of that. And importantly, when we label the cells with biotin and then probe the cell lysates for streptavidin HRP, we can detect biotinylated proteins. So we enriched those with streptavidin beads and used mass spectrometry to identify the proteins with 3 independent replicates across each condition. And what this PCA is showing are across the replicates, the similarities of the overall protein profiles that were identified, showing clustering of these different transgenes with the different replicates, which is indicative of really cool and high quality data, as further represented by this heat map showing the proteins, each row is a protein that were found in each of these different complexes. And so all these proteins, we can perform gene protein enrichment analysis with known protein complexes, and we can find interacting partners of CDK12 that are both known and novel, including those that are known in spliceosome or small nuclear ribonucleoprotein complexes, or other interesting unknown complexes. It's quite a sensitive and robust approach to identify protein interactions. And I just want to note that we've found some interesting ones that we're pursuing, including interactions with the RNA modification machinery, as well as other chromatin complexes to better understand the role of CDK12 in these biological processes. This should say AIM-2. So AIM-1, we were able to establish our protein-protein interactions. Our next approach was to study the protein using a Dagron approach, where we can do acute depletion of CDK12. And the way the Dagron works is we wanted to generate cell lines using a CRISPR-Cas9 system, whereby we target the genome using CRISPR-Cas9 to make a double-stranded break, and then the presence of a homology-directed repair template containing a piece of DNA that encodes an epitope tag and a destruction tag. We can tag the endogenous CDK12 loci such that it expresses this tag. And so then cells that express this tag, and this is the only CDK12 version in these cells, are very sensitive to treatment with this chemical called DTAG. What it does is it induces ubiquitination and protein turnover rapidly of this CDK12 protein. So we sought out to establish these Dagrons in HER2-positive breast cancer cells, and using this methodology of CRISPR-Cas9 and knock-in of this piece of DNA in frame to generate these constructs, and then apply this to various genomic or proteomic methods to study CDK12 function. And I should say that we were a little naive to try to be able to generate this in HER2-positive breast cancer cells because of copy number. This process of generating knock-ins in the genome is challenging when you have dozens of copies of a gene. So we had to do a proof of principle in a cell line called HEK293 cells. These are human cell line. And this is showing some of the data of the Dagron. I'll walk you through the results really briefly. So these are the Dagron. This is a clone 4 of this Dagron. We selected several clones. And this is at time 0 with no inducer of degradation. And we can see when we probe for CDK12 or the HA epitope is that the cells express this protein. However, within 10 minutes or up to 24 hours, with induction of this DTAG protein, we have stable turnover of this protein and complete direct depletion of CDK12. And in these lanes, we're comparing parental HEK293 cells treated with CDK12 inhibitor. And what we can see from these studies when we probe these proteins, CDK12, one of its main substrates is RNA Pol2 at phosphoserine 2. It generates phosphoserine 2. And we were surprised to see at 10 minutes of degradation in an hour that this level of this substrate of CDK12 actually went up, because this is a described target. So this just further highlights the complexities of these problems. And what we know is based on a lot of coordinated and difficult to disentangle experiments. And so we hope this Dagron will help provide a direct and analysis a system where we can study some of these fundamental transcriptional mechanisms. What this plot on the right is showing is an analysis of live cell imaging cell growth of HEK293 cells treated with a control, and they keep on growing, but those treated with the D-tag or the inhibitors fail to grow. So indicating that this system has a consequence on downstream cell growth and highlighting CDK12's role in this process. With this system, we were able to do a few cool experiments where we could induce degradation of CDK12 and measure nascent RNA transcription over time using a protocol called SLAM-seq. It's metabolic labeling with sequencing. And what this volcano plot is showing is that when we induce the degradation of CDK12, we can really turn over newly synthesized RNA genome-wide, and that CDK12 has an important role in RNA transcription. And that importantly, that CDK12 destruction results in a higher fraction of introns, which is supportive of its role in RNA processing and splicing. And that when we destroy CDK12, we see interesting genes such as those encoding the U1 spliceosomal RNA are downregulated, which might explain some of these splicing effects. And these are sort of novel findings using this system. And furthermore, we have a way of performing ChIP-seq to map the binding sites of proteins genome-wide with CDK12 depletion and where we can look at RNA polymerase genome-wide around the transcription start site of all genes. And what we see is an enrichment of RNA polymerase around the promoter or transcription start site of genes as expected that doesn't change with CDK12, and that these profiles along the gene body to the transcription termination site is lower. But when we look at phosphoserine 2 profiles, we can find differences. We can find genes that don't change with CDK12 depletion, but we find selective regulation over genes with treatment of CDK12. And that mostly manifests itself as either being those with increased serine 2, which is consistent with that Western blot we saw, but also genes that are down. And so this is totally unknown, and we are excited to further study this. And so I'm just going to summarize now. AIM-1, we were able to establish CDK12 cyclin K interactomes. We're actually working on CDK13 so we can have the full complement of both of these related kinases. And we're going to validate these via co-immunoprecipitation and proximity ligation assays to pick some of these candidates and study them. In particular, we're interested in the nuclear organization that the role that CDK12 plays in nuclear organization and RNA modification. In fact, I just submitted an NIH NCI R21 grant based on this preliminary data to study CDK12 to modulate M6 RNA modification. Just briefly, M6 modification is turned out to play a key role in cancer and HER2 positive breast cancer as well. And targeting it is not so easy. And so if CDK12 can function to regulate M6A through its kinase activities, that could be an important intervention for targeting RNA modification in cancer. We also aim to establish the Dagron, albeit in 293 cells, not breast cancer cell lines. We're not giving up. This shows it. We just need to be keep working on it and get lucky. But we're also interested in generating a double Dagron to study the functional redundancy between these two proteins. And then furthermore, to study the substrates of CDK12 and 13, because truly their validated substrates are very low of all human kinases. And so applying this Dagron model and then performing temporal phosphoproteomics would be very powerful to study its role in nuclear signaling. All right, with that said, I want to acknowledge the key players. Hannah in particular has done all the work, and I was all the behind the scenes on all of this. Peter Kaufman, who has been key for helping us derive clinical meaning out of some of these studies. And then I want to thank Janet Stein and Gary Stein, Fran Carr, Doug Tachi's the member of the MIC Vigor Corps at CBSR, who have all been so important for me and these project developments, and then my department at UVM. With that, I'll take any questions.
Oh, great. Thank you, Seth. That was very informative, very nice. Good molecular and proteomic biology there. So if there's any questions from the group, this is a good time. We'll take just one or two questions and we'll move on. Any questions? No? Well, I do have a question, Seth, and that is, you had a figure that sort of represented the mTOR pathway along with your CDK12. Is there any connection between CDK12 and mTOR?
Yeah. So absolutely, good question. mTOR is a key downstream target of the HER2 signaling pathway, actually. PI3 kinase, mTOR are activated through these pathways. And we hope by studying CDK12 in HER2 positive breast cancers that we might actually be able to understand the connections between those downstream signaling pathways and CDK12. If the idea being that if we can target CDK12, we could cut off those cancer dependencies directly.
Right. Yeah, because it's involved. That's highly involved in nutrient signaling.
Mm-hmm.
So yeah, it's kind of a big deal. It's a
Yeah.
really, yeah.
So the gene expression profiles and even potentially the RNA modification profiles might be helping drive those nutrient signaling expression patterns that the downstream patterns, and so they're related to that.
Yeah. Okay.
Yeah.
If I may, Rob.
Oh,
Roberto.
yes, Roberto. Yep, yeah, please.
Hi, nice meeting you, Seth. Very nice work, beautiful. So I have just a question. When you show that the use of the knocking down the expression or adding inhibitors of the CDK12, um, was that a cell growth arrest or a cell death? And have you tried anyway your approach to see a potential eventual a therapeutics application of the approach?
Yeah, thank you for the question, Roberto. Yeah, the image I was showing was based on some live cell imaging. So you just seed the cells, add the compounds, and then watch the cell growth. We haven't done much phenotypic characterization of that. That would be very interesting to see, what happens to cells over time with inhibition. With therapeutic applications, truly this is a competitive field. A number of groups and companies are targeting this molecule, and which is great. So we haven't done direct therapeutic applications, but we have some selective inhibitors, and we're comparing our genetic studies to those inhibitors. And so those inhibitors could be working through off-target effects, and also in our protein-protein interactions, we hope to find things that are additionally related, and then get a clinical angle from a therapeutic angle from our basic studies.
Yeah, I mean, it was after what you showed is a pretty broad number of targets you're
Yes,
having with that. So
it's a nice data set, but yes.
yeah. Okay, very, very, very beautiful. Okay, love it.
Thank you.
Thank you.
Okay.
right. We'll move on to the next speaker. Thank you again, Seth. Very nice presentation. And so next speaker is Liz Scharnetzki. She's a member here at the Maine Health Institute for Research in the Center for Interdisciplinary Population Health Research. We call it CIPHER. And she is a experimental social psychologist, has lots of experience in research design and implementation, survey methodology, as well as advanced statistical data analysis. And she'll talk to us today about her pilot project entitled Adapting Boot Camp Translation Methods to Engage Rural Communities in Lung Cancer Screening. Okay, Liz, welcome.
Appreciate it. All right, let's see if I can do this with finesse. At this point, right, we should be all... How does that look for everyone? Look all right? Wonderful. All right, so as Rob mentioned, my name is Liz. I'm a faculty scientist and experimental social psychologist at Maine Health Institute for Research, and I'm so pleased to share some study updates with all of you from our pilot project. At the offset, I wanted to give a big acknowledgement and expression of gratitude to Erica Biot, who is really been our community champion and my co-facilitator in this work. And of course, Dr. Neil Corson, who many of you on this call know, who has been my mentor on this project. I'll also mention that we did receive a no-cost extension for this work, so we're not quite done, but we have a lot of great progress to share with you today. And of course, a huge thank you, NNE-CTR, for the opportunity and support to be able to conduct this work. And why are we doing this work? Really, the long and short of it is that the burden of lung cancer in Maine is very significant. Lung cancer is the third most common malignancy and the leading cause of cancer death in the US, but here in Maine, our incidence and our mortality rates are significantly higher than these national averages. This is in part because the majority of the cases here are diagnosed at a late stage, but it's also a result of intersecting factors that include Maine having a high rate of tobacco use, high levels of environmental radon. We also have a large population of lower income rural residents who experience significant systemic barriers to accessing care and who feel stigmatized and disengaged from our healthcare system. It's not all doom and gloom, though. There are many reasons to be very optimistic about the future of lung cancer care. In particular, our early detection options have undergone tremendous scientific advancement. Low-dose CT screening, for example, for the early detection of lung cancer has been shown to significantly reduce mortality among eligible high-risk individuals. And low-dose CT screening has been recommended by the USPSTF since 2013 for lung cancer. Unfortunately, though, here in Maine, our screening rates among our eligible population remain pretty low. They've been reported to be as low as 9.7% of our eligible population being screened, which is lower than many of the other northern New England states. And that's a significant problem given the fact that the burden is so high in our state. So really providing our patients, and particularly those in rural areas, with these early detection options for improved prognosis needs to be a priority. So what are some of the factors that could be driving the lower uptake of early detection screening for lung cancer? We have heard in other work from our communities that lower income rural Maine communities feel disengaged from the healthcare system due to a history of being stigmatized and medically underserved. And in fact, this is supported by a lot of the stigma research that has identified stigma as one of the most prominent barriers to equity in the detection and treatment of lung cancer. 95% of patients with lung cancer report experiencing stigma, which actually makes lung cancer a more stigmatized cancer than all other cancer types. For those who aren't familiar with this body of work, social sciences define stigma as a mark of disgrace that's associated with a particular characteristic, quality, experience, or identity that our society devalues. And lung cancer stigma is a form of health-related stigma, in which a person perceives and maybe even internalizes the belief that people perceive their behavior as having caused their current health condition. So in the case of lung cancer, the association between smoking and this condition is very, very high. The internalization of this kind of stigma can lead people to feel things like shame, blame, fatalism, and nihilism, which I'm sure you can imagine doesn't have amazing consequences. And in fact, it does not. So stigmatization has been shown to be associated with poor patient-provider communication, constrained disclosure, a delay in detection and treatment among low uptake of other recommended health-promoting behaviors, poor treatment adherence, all of which really culminates into disparate health outcomes for lung cancer patients relative to less stigmatized health conditions. So another thing we need to consider then when we're trying to engage our eligible population in early detection is to avoid perpetuating or evoking this kind of unintended stigma. The other factor that could be driving these really low screening rates is our translational science process. So I don't really need to tell this audience this, but here on the slide, you'll see our translational science spectrum moving from bench and basic research to bedside practice-oriented outcomes. And unfortunately, however cool this is, it is a very, very slow process. It's about 17 years if we want to, on average, if we want to get really exact. So it's a very slow process. And addressing this barrier and promoting the more effective translation of science is really critical if we want to engage people in this. Luckily, there's a group of people who are on the job and on the problem, and there's evidence to suggest that community engagement, and in particular, the use of an approach called bootcamp translation, is a really successful strategy to optimize the translation of medical and scientific evidence and get it out into community in a lot quicker than 17 years. All right, so what is bootcamp translation? It is an innovative community-based participatory research approach that alters the typical conceptual framework for communicating scientific evidence and information. Diverse stakeholders, so rural clinicians, community-based organizations, community members are engaged together to co-develop solutions for a local health issue. Stakeholders and researchers come together to share their personal lived experiences, knowledge of how their community receives information, what kind of community resources are available, and of course, research expertise to develop more locally relevant and ultimately more actionable messages for a targeted subset of members of their community. Because BCT uses this high degree of community engagement, it's actually been shown to be more effective in producing better health outcomes compared to other strategies, whereas more traditional efforts to engage particularly rural residents rely on a more rhetorical dissemination of clinical and scientific evidence and have largely failed. BCT developed messages have been shown to engage more than 1,000 rural community members across many different states and have demonstrated improvements in care for a wide range of health conditions. In one particularly relevant BCT project, which aimed at promoting colon cancer screening in rural Colorado communities, they showed that BCT developed messages increased their community screening rates by up to 10%, which is a pretty profound jump. So how does BCT work? Traditionally, it's a 3-phase process. You'll see here I have a 4th phase, which I'll get back to in a moment. But essentially, BCT teams are comprised of an ensemble of about 10-ish people, academic researchers, subject matter experts, and of course, community members. Each team has a community champion or community connector. Ours is Erica Biot, who's on the call here today, who co-facilitates the entire process with the research study team to ensure equal participation and shared leadership. So the 4th phase you'll see I added here at the top is building community. We added this in because we felt it was important, especially in our new kind of hybrid world, to spend some time building community and rapport with one another, separate from kind of the official research phases. So we did this by hosting an in-person kickoff. We shared food, we had conversation, we got to know each other, and we plan to host a wrap-up gathering as well to celebrate the completion of our BCT. After that point, we've been following a very typical BCT approach. Phase 1 includes education sessions on the health topic area that are facilitated by subject matter experts so that all BCT members can work with a common and shared understanding. In phase 2, the team engages in facilitated conversations to answer 3 questions. What's the focus of our message? Who is our intended audience? And what are we going to be advocating for? And then in phase 3, members of the BCT group actually work together to develop the messaging intervention and the plan for disseminating the messages out into the community, as well as designing the evaluation of how we will determine whether our intervention was effective or not. So what we wanted to do in this pilot project was to adopt and use this methodology to develop a rural-focused intervention for low-dose CT screening, with the ultimate goal of reducing some of the disparities in low-dose CT screening access and utilization. Specifically, we really wanted to target the knowledge gap, that translational barrier, as well as maybe combat some of the negative attitudes and stigma-related beliefs around low-dose CT screening and lung cancer. So we had two specific aims. The first was to develop and implement this communication-based intervention for low-dose CT screening using the BCT method, which to our knowledge, no study has done to date. So this is the first to use BCT for lung cancer screening. And the second aim was to assess the impact of our BCT developed messages on our target community's awareness of and attitudes towards screening. And we hypothesized that relative to baseline assessments, awareness and attitudes would improve in the community in which our messaging intervention was implemented. And again, to accomplish these aims, we did everything in very close partnership with Healthy Community Coalition of Greater Franklin County in Farmington, Maine. Erica Biot, here today again, served as our community co-facilitator through the entire process. And I am not overstating that nothing would have gotten done without our very, very close partnership and collaboration with this team. So I can't say thank you enough. All right, so in phase 1, we hosted 4 education sessions and an interdisciplinary group of subject matter experts. Our first session was with Dr. Neil Corson, who kicked us off and gave a thorough overview of lung cancer, cancer screenings, low-dose CT screening in particular. And I would say the health information was delivered at about the level of a medical school education. Next, we were joined by Dr. Jamie Stutz, who's a clinical psychologist and the scientific director of behavioral oncology at UC Denver. Dr. Stutz has expertise in the psychosocial factors that are associated with low-dose CT screening implementation, and he spoke to our group about the stigma, nihilism, and fatalism that can be experienced by folks who are at risk for developing a smoking-related illness, and how we can move towards a model of more person-centered care and promote equity and availability and access to low-dose CT screening. Our third session was hosted by Dr. Danelle Boatman, who joined us from the West Virginia University Cancer Institute. Dr. Boatman is a health communication researcher who focuses on cancer communication in particular, and she shared with us some of her research on lung cancer communication strategies that they have been testing to promote engagement in screening in Appalachia, which is, of course, another rural and historically medically underserved area. So we had a lot of great lessons learned from Dr. Boatman. And lastly, Dr. Dawn Neese wrapped us up. Dr. Neese is a professor of family medicine and the director of community engagement and health equity at UC Denver, one of the pioneers of boot camp translation. And so Dawn gave us a refresher on the methodology, the philosophy, and shared some of his group's previous BCT work so to serve as some inspiration as we move into our second phase. In our second phase, we had facilitated conversations where we reviewed what we learned in phase 1 and worked through the 3 questions on the screen. What does our community need to know about lung cancer screening? Who in our community specifically needs to hear this message? And how do we get the message out? So you can see here, this is just a screenshot of some of our brainstorming sessions. We created a virtual whiteboard or bulletin board, put up all possible ideas and then sorted on them, voted on them. And we did this many, many, many different times. Lots of fantastic ideas. And then on our third phase, we worked with Rink Advertising, which is a local advertising firm, to develop messages. So I'm going to show you 2. They are still in draft form. So we just sent back some edits and feedback for Rink to make some final tweaks and changes, but we're really excited and really thrilled with how they're looking so far. So these are drafts, but here you can see 2 of the messages that we've been working on so far. So the first one is things you can do while wearing your jeans. If you didn't know, you don't have to do any kind of scary prep for low-dose CT screening for lung cancer. You can go in your street clothes. You don't even have to put on a gown. But when people hear lung cancer screening, there's a lot of anxiety, and it even has a term associated with it, anxiety. So to combat some of that stigma and fear around it, we tried to kind of think of what could be a little bit more lighthearted approach. And again, very locally and culturally relevant. This list here, I know it's small, but it's all things that you can do in the Farmington area, of course, while wearing your jeans, like going to the Farmington Fair, walking the rail trail, visit small falls, and then of course getting screened for lung cancer. And then at the bottom, it has information about how you can talk to your doctor, eligibility, and where you can get screened for free if you are eligible. Similarly, in the second draft message here, we wanted to combat some of the fatalism or nihilism and just take a more optimistic approach to messaging. So we created a montage of images that could represent the different generations that may or may not be eligible for lung cancer screening, created sort of a little game, which of these messages or which of these images describe you. And if you select one, you know what, it might be time to get screened for lung cancer. And then similarly, we provided information about how they can talk to their doctor about it, eligibility, and the fact that they can get lung cancer screening for free if they are eligible, and information on where they can do that. All right, so we are conducting a two-phase evaluation of this pilot, and phase one is focused on evaluating how well our BCT process has gone with our group members. So we will conduct 3 different time point surveys. We've conducted 2 so far. So we are doing a baseline, a midpoint, and a final survey. And we're assessing constructs like engagement, psychological safety, shared leadership, perceived value, knowledge, and perceived impact. And I'm going to share a little bit of the preliminary data now. So from our baseline survey, or before I go into the baseline survey, I should say that our BCT group is relatively small. We had around 7 to 8 regularly attending members, and some folks opted out of evaluation. So hold the caveat in mind that our ins are small, but mighty nonetheless. All right, so from our baseline survey, we wanted to get an idea of our BCT group members' baseline subject matter knowledge. So we asked about things like their familiarity with risk factors associated with lung cancer, how familiar they were with the concept of lung cancer stigma, their understanding of low-dose CT screening, as well as some measures of health communication and health messaging in their community. The take-home message here is really that, in general, our BCT member subject matter expertise was already pretty high. We also asked our BCT group members in this baseline assessment about common perceptions and sources of health information in their community, as well as how health information gets out. Really important for us to kind of know how information is circulated and operates. And what we found was that public health officials appear to be very key. And while some of the community is reached by current health communication efforts in this community, there's certainly a lot of room for improvement. We also asked people about their expectations for the BCT process, and here's just a selection of some of the illustrative quotes. What did they hope for? More knowledge about lung cancer and to be part of educating their community, what their desired outcome would be as a result of participating in this BCT process, so increased community knowledge. Personal hope, again, more knowledge. So you can see it really seems to be a trend, which is great because that was the ultimate goal of our BCT, was to increase knowledge and positive perceptions of lung cancer screening. All right, so I'm going to transition now to share some preliminary results from our midpoint survey data. So we asked people about the degree to which the BCT process has increased their lung cancer knowledge and their low-dose CT screening knowledge. And granted, while our group's baseline knowledge was already pretty high, this BCT process did appear to support increased subject matter expertise. So it does appear to at least sustain knowledge and add to participants and members' existing knowledge base. We found that our BCT members experienced few barriers to engaging in the BCT process, which was excellent. And in general, they reported feeling that the BCT process had been proceeding at a high quality level and was very valuable. So that's good. We were respecting people's time and using it well. We also asked people about what their experience has been like so far. Did they feel their voices were heard? Did they feel safe, psychologically safe, engaging in the BCT process? Did they feel respected? Did they feel like they were getting a good understanding of the BCT process, not just the health topic area of lung cancer, but do you feel like you're gaining expertise in BCT? And in general, you can see here that our ratings were really high. So that's wonderful. Seems that people had overall a really positive experience with the BCT process. This is a really big graph, so I apologize in advance, but I'll try to unpack it relatively quickly. At the midpoint survey, we also asked, similar to the baseline, about whether or not the BCT process was supporting their learning and understanding of both lung cancer related topics and health messaging. So again, things like understanding of lung cancer screening, stigma, familiarity with risk factors, low-dose CT, and the relevance of this kind of work to community health messaging. And you can see overall, with the exception of a couple of the community-specific data elements, that people had pretty positive perceptions of the BCT process supporting increased knowledge understanding across these different dimensions. And then a couple more illustrative quotes when people were asked to explain the value of BCT, working as a team to gather information that is relevant to our community members, and then perceived impact on the community from this kind of a project, and specifically the BCT messaging that gets developed as a product of the process. It will get people thinking more about their health and screenings that are available to them, which is excellent. Once again, spot on, that's the goal. So our phase 2 evaluation is going to be focused on evaluating the impact of the messages that our group has developed together. So specifically, we're using a pre-post design where we have co-developed a community knowledge survey that assesses our target communities' knowledge of lung cancer screening, their attitudes and awareness towards lung cancer screening, and in the post survey specifically, whether or not they were exposed to our BCT developed messages. And as I mentioned before, we have co-developed everything together as a BCT group, including our community knowledge survey. So we have developed our survey. It is under IRB review right now, so that's really exciting. Our next steps are to disseminate our baseline survey, our messages, and our post survey, and develop kind of a sustainability plan, because we would love these materials and this work to live on in this community and any other communities that could be interested in utilizing this kind of information or tool to disseminate information. But in general, our overall hope and the future directions of this work is to use this as pilot data to be able to apply for, of course, subsequent funding. We are very interested in the future question of how local is local. Can we scale this up? Can we adapt this? Can we look at the impact of these kinds of BCT developed messages in multiple communities? And then, of course, another really, really important future direction for this work that I feel this pilot project can serve as a springboard for is moving away from assessing the impact of BCT developed messages on knowledge and awareness and attitudes, which of course is very important, but actually measuring things like screening rates in the communities in which these messages have been implemented. And like I said before, all the credit goes to our team. Here's a Zoom picture of our community team at one of our last meetings. So I just can't express enough gratitude for their sustained engagement through this pilot project. We are so lucky. And I'm happy to take any questions that folks have.
Okay. Thank you, Liz. So Ben, you have a question? I see your hands up.
Yeah. That was an awfully nice presentation. I'm interested in how you're going to get the messages out there. How did you pay for the PR firm, and how are you going to pay for the publicity?
Yeah, that's a really great question. So part of our budget, and we've already been working with Rank, went to working with a local ad firm on not only the development of the messages, but also the strategies and making sure that our messages could be adaptable across multiple different platforms. And then in terms of how we're getting it out, that's actually something our group has been working on together that we are tackling and doing. So we have identified a few different places that we are going to disseminate our messages. The bowling alley has screens that are going to potentially display and project the messages. There are local virtual newspapers that will get digital copies of this. We have adapted versions of these messages for social media that will run on all of our partner organization sites as well as our individual sites. And then we have flyer versions of these, which will go to community centers and hubs as well.
So no media costs.
There were some that went to Rink, but Rink has really been kind of a all-inclusive hub.
No, I mean TV, radio, newspaper ads.
Yeah.
Out of your budget.
Out of my budget. I mean, I'll take more money, but.
Good work.
Thank you so much.
Yeah, Gary?
It's a beautiful presentation. I mean, an example of, the power of community engagement. To try to go ahead and persuade folks to go ahead and address prevention. Question I had was, are you planning to be able to take the lessons learned from the community that you've worked with and try to look at it in other communities so you can assess how, in fact, the shared unique characteristics of the different communities might impact the particular persuasion approach that's developed?
Oh my goodness, Gary, don't scoop me. That's one of our future directions, most definitely. So we've been lucky to connect with some folks that are very well versed in the BCT method and process in other areas. And so we are very interested in being able to examine actually the adaptability of these messages and understand what translates across communities and what specific elements really need to be locally edited and tailored. So I think that that is a really important next step that's not been done, to my knowledge, in the BCT program of research yet, but it's certainly first on our list.
Thank you.
Great, thank you. Any more questions for Liz?
Just one comment. You may already know, but a lot of what you're talking about just completely resonates with the Patient-Centered Outcomes Research Institute. Mm-hmm. So it's probably time to start nosing about there for funding.
Thank you. I will definitely appreciate that. Thank you, everyone.
Great. I think, yep. So I think that's good. Thank you, Liz. That was a great talk. So next week, I think we have another set of presentations. I think we're done for today. Sheila, you would like to pitch in and just tell us what's coming up next week on the docket?
Yeah, I just posted in our chat we have 3 more pilot presentations next week, and then the following Monday will be a ChatGPT seminar, and that'll wrap up our spring series. So it's been a robust series this spring. Next week, we'll have Dave Kaminsky on feasibility of home monitoring of lung functions and symptoms in patients with COPD exposed to wood smoke at home, which is a very relevant topic here in the Northeast. We'll have Connie Van Egan talking about opioid prescribing patterns and analysis of secular trends. And Tim Burdick from the PCBRN will be talking about a comparison of low-burden data collection methods using a card study on primary care, telehealth, and burnout. So I'll send out that email blast with the Zoom link and the flyer on that this afternoon. And hopefully we'll see you all next week.
Great.
Thank
Okay.
you.